personalized medicine. Use molecular markers to tailor treatment according to that patient. So that patient, although both of them may have lung cancer, lung adenocarcinoma, this one will get different treatment than the other one because he has different mutations. واضح?
Yes.
So personalized medicine, you tailor the treatment according to the patient. Two patients has exactly the same diagnosis of lung adenocarcinoma, but they get a different treatment because of the different molecular profile, molecular testing results. So we call it personalized medicine. You give it according to the patient. And this is the new concept of treating cancer. So the new concept is personalized medicine. You give the patient according to the molecular profile. So you have to do molecular testing. Any question on this? This is microarray study. Did you study microarrays?
No.
Okay, microarrays is just introduction. You don't have to remember it, just for your information. It won't come in the exam. Microarray is this is the new way of personalized medicine. We know each tumor has its own. As I mentioned to you, you have 10 patients with lung cancer. Each one has different gene expression. So we studied the gene expression of tumor by comparing normal tissue of the patient with the tumor tissue. So we are studying the gene expression. So how we study, what are the gene expression? What we have to test? What we have to study gene expression? Either messenger RNA or protein. The DNA, it doesn't mean that it will be expressed. صح? You have to study messenger RNA or protein. To study gene expression, you study messenger RNA or protein because the DNA itself doesn't mean that it will be expressed. So in microRNA, we study messenger RNA of the patient, but we need fresh tissue because messenger RNA is degraded quickly. The DNA is not degraded. من أيام الفراعنة الDNA ما في مشكلة بس المسنجر RNA it will be degraded in 30 minutes, 1 hour it will be degraded. Why? Because the function of messenger RNA is temporary function. It goes from DNA to protein actually degraded. واضح؟ The function of messenger RNA should be short-lived. It's just transmit the message from DNA to the protein, to home protein. And it should be degraded by RNA. Whereas DNA is there. If we want to study messenger RNA, we have to have fresh tissue and do it quickly. We have to extract the messenger RNA quickly. Otherwise, we lose it. DNA, no. It takes long time. So, you have normal tissue, you have tumor tissue, you extract them, and you compare hundreds of the genes. So you need bioinformatics. You have special software, you have special person specialized in bioinformatics. Not anybody can do it. Special training to analyze the gene expression of normal tissue and the tumor tissue and see the difference. Then we see what are the tumors that express in the genes that express in tumors, not expressed in normal tissue. What are these? Then we can study those genes, focus on them, and develop treatment. Do you understand?
Yes, sir.
That's the concept. So, so the reason to study this hundreds of genes to see what are the genes that are expressed in tumors, and we focus on them and develop. They become either diagnostic or prognostic or predictive.
The red one means that the person
The red? No, the red is overexpressed. The red, you have 3 colors: red, green, and black. The red means overexpressed, green underexpressed, black means not expressed.
So why are we hybridizing?
Sorry?
Why are we hybridizing it? Like we're mixing both the...
No, we hybridize the tumor alone and the DNA, the tissue alone, and then we see the expression by bioinformatics and see which one is expressed. Which one expressed in normal, same patient, normal and tumor tissue. And the goal to see which genes that are expressed in tumor, not expressed in normal tissue. To focus on them. That's it. Typical example that we use gene expression nowadays for treatment, again, you don't have to do that, in breast cancer. We have mammogram and oncotype. These, we are using 70 genes. In breast cancer, you have a patient who has no lymph node metastasis, Q1, this is a vaccinator. We don't know which one will develop metastasis in the future, which one no. So we do gene expression to know. If that patient will develop metastasis, we'll give it chemotherapy. If not, we won't. So the gene expression tells us these patients will develop metastasis, so they will give them chemotherapy from the beginning. Only small percent. We are speaking about non-negative patients, or not. The goal of treatment is cure or palliation of cancer patients. The number of patients to be cured from cancer are getting more and more. But palliation is not cure. Is to improve the lifespan of the patient. This is the treatment principle: biological behavioral tumor, extent and bulk of disease, mortality and morbidity of therapy. This is just for reading. Now, screening. We said 2 types of screening. Early diagnosis is preferable, like breast cancer, or diagnosis at pre-malignant stage, like cervical cancer. Prevention. Behavioral modification, smoking, sun exposure, this is the treatment possible. Risk avoidance, occupation risk, dietary factors. That's the principle of treatment for cancer. Now, at any stage. Now, we have 2 types of therapy. Exploit the properties of tumors, professional dose of cell cycle control, genetic stability. Most drugs and radiation causes massive DNA damage and induce cell cycle arrest and cell death. So, chemotherapy and radiotherapy act on protecting cell in the cell cycle, right? And so it doesn't differentiate between tumor cells and normal cells. That's why the patient has hair loss, GI abnormality, any cell, bone marrow is affected with anemia, because any cell that is dividing in cell cycle will be affected by therapy. To avoid that, we have new class therapy, what we call targeted therapy. Target the cells that have the hemoglobin. And we gave example like EGFR in lung cancer, it was 20% of patients. If you give them treatment, they target the lung cancer, and that's why they have this side effects. Than chemotherapy. And Herceptin in breast cancer. So target therapy have this side effects because they target the tumors. They have some side effects, not pure targeting the tumor, but they have this side effect like chemotherapy and radiotherapy because chemotherapy they target everything. Any cell says this inside.
Yeah, but why not start just giving the targeted one instead of the general chemotherapy?
How do you target it?
Target, you have to have a target. We said that. We say that if the patient has no target, you can't do target. What's the target? In patient who has breast cancer, you have to have HER2 amplification to do the target. If she does 80% of breast cancer, they do not have target therapy, HER2 amplification. So you can't do the target therapy. واضح? Is it clear? 70% of lung adenocarcinoma, they don't have EGFR mutation. So you can't give the target therapy. And target therapy is very expensive. It has side effects, but much less than chemotherapy. It's more specific. So we have chemotherapy, radiotherapy, and target therapy. The problem with chemotherapy and target therapy is resistance. So all therapies like bacteria, just like bacteria and antibiotics, resistance. Why resistance, you think?
Mutation.
Tumor cells are killed. Once you give the chemotherapy or target therapy, they change the mutation. So breast cancer, they depend on HER2 amplification for their survival. So they don't have survival. So we give Herceptin as amplification. They die. Then as new clone of tumor cells, they develop new mutation for the survival. Then a tumor regrow again. And that's it. So development of new mutation. The cancer soaks it level. You have EGFR, you give him Tarceva as therapy. Then those patients will respond for a few months. then a new clone comes, there's no mutation. So they want to swap faster. So that's the problem with the resistance, like antibiotic. واضح هذا؟ If the uptake pathway is not working, then drugs will not work. Therefore, increased resistance when there is mutant p53. Increased expression of multi-drug resistant in tumors. You monitor cancer by radiology, clinical exam, and so on. So this is the last slide I want you to remember. Last slide. Now we have biomarkers. There are hormones that we can use as biomarkers for tumors. شباب ركزوا معي. This is the last slide. HCG, human colonic gonadotropin, in colon carcinoma, calcitonin, in medullary carcinoma of thyroid. Ectopic hormones, oncoprotein, alpha-fetoprotein in liver cancer, CEA, carcinoembryonic antigen in colon cancer. No, I want you to know only the hormones and the oncoprotein and PSA. PSA PSA here. PSA. So what I want you to know from the biomarkers, I want you to know the hormones, and the oncoproteins and PSA. PSA, prostate, specific antigen, and prostate cancer. These are the
Want us to know all the oncoproteins?
yeah, oncoproteins, alpha-fetoprotein, and CE.
All of them.
So oncoprotein is two. And hormones. Hormones. And PSA. PSA in prostate specific. Hormones, oncoprotein, alpha-fetoprotein, and CEA, carcinoembryonic antigen, and PSA. PSA here. A prostate specific antigen in prostate cancer.
The rest we don't have.
You don't have to. Any question على النيوبلازيا؟ واضح النيوبلازيا؟ هذه الأسئلة. All questions.
We have to memorize all this thing.
No, I said the hormones, oncoprotein, oncofetal protein, and PSA, prostate specific.
أقول لك بعد.